AP 120, Chapter 8, Topics, Myogram and Muscle Tissues. All right, so a myogram is basically a graph showing the amount of force generated by a single muscle twitch, generated by a single muscle fiber contracting. Although, to be honest, that's usually using an entire muscle because that's just a lot easier for the research. And then this part, the x-axis, as it were, is just the passage of time. So we got the force of contraction and the passage of time. And what we find is that for a single muscle twitch, you need a threshold to be reached. So basically what they do here is they would give an electric jolt to the muscle. And if the jolt isn't powerful enough, then nothing happens. However, if it is a strong enough electric jolt, then you reach threshold and you will get a muscle contraction. After the stimulus, there's a slight latent period. It takes a little time for the muscle fibers to respond and contract. Then you have the contraction period in which the force of the contraction gets greater and greater and greater until it maxes out. And then you get the period of relaxation as the muscle fiber relaxes and returns to its original length. And again, this is an all or none response. Either you reach threshold and get a contraction, or you do not. Now, it is of course possible to zap that muscle multiple times. So here, the person just zaps it, gets the contraction, waits for the muscle to relax, zaps, contracts, relaxes, waits again, etc. Just boom, 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 boom. And you get the complete relaxation between the multiple twitches. Turns out, though, that if you zap the muscle again before it's completely relaxed, you can actually increase the amount of force generated by that muscle. So you zap the muscle, it contracts, then as it starts to relax, you zap it again, it contracts, but now it generates even more force. Zap it again, it contracts and generates even more force until eventually you let it relax again. And this is called summation, the increased force of contractions by applying stimulus rapidly and enough. Then you have complete tectonic contractions. Complete tectonic contractions is when you zap the muscle so fast that it has no time to relax and reaches the maximum force possible, the maximum amount of force that that muscle could possibly generate. So again, you zap it, you get your contraction, you zap again and again, you zap faster, 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 and then eventually you get complete tectonic contraction. The entire muscle, all of its muscle cells contracting in unison, generating the most force possible. All right, so how do we get those different amounts of force generated by a muscle? Well, it's because the muscle has many motor neurons going to its many muscle fibers. And so it just has increased signals from these neurons to allow for increased amount of force, basically recruiting more muscle fibers to contract and generate that force. So we have the concept of a motor unit. A motor unit is a motor neuron and the specific skeletal muscle fibers that it controls. The motor unit is one motor neuron and the muscle fibers it controls, and all those fibers will contract in unison when the nerve impulse travels down the axon of the motor neuron. And you have multiple motor units within a single skeletal muscle. So when the muscle needs to contract with more force to pick something up even heavier, then you have recruitment, which means more motor units are recruited, activating more skeletal muscle fibers, leading to a more powerful contraction. So we can, if you're, say, lifting a piece of paper, you might only have 10 motor units activated in those muscles, but when you try to pick up a textbook, you might have 20 motor units activated in your muscles, and so on. Muscle tone. Turns out when we are awake, our muscles are always continuously slightly contracted. So there are always a few motor units contracting and active in each of our muscles when we are awake. And this includes the muscles for posture. And so our posture, the muscles controlling posture, are always active, holding us upright. Whether we have bad posture or good posture, there are muscle contractions involved in that. All right, moving on to muscle tissues. This should be familiar from Unit 1. Skeletal muscle tissue. Skeletal muscle tissue is what's found in our skeletal muscles. Surprise, surprise. Usually these muscles are attached to bone and controlled by conscious effort. So skeletal muscle tissue contracts via voluntary control. We choose to contract those muscles most of the time. The cells in a skeletal muscle tissue is extremely long. These cells can be the length of the entire muscle. So if you have, say, your biceps muscle, biceps brachii, that muscle is what, maybe a foot long. So that means the cells within it are about a foot long. These cells are also cylindrical in shape. As we know, they're striated, so they have that dark light pattern formed by the sarcomere, as we know. Here, down here's a little sarcomere. Light, dark, light, dark, light, light, dark, light, dark, light, dark striations caused by the sarcomere. Also, skeletal muscle fibers have many, many nuclei, tens, 20s of nuclei within one cell. And of course, they contract in response to a nervous impulse. Then we have cardiac muscle tissue. Cardiac muscle tissue found in the walls of the heart. Cardiac muscle tissue is similar to skeletal muscle tissue in that it has striations. So again, we have that dark light pattern going along the length of the cardiac muscle fibers. So that, of course, means that cardiac muscle cells have myofibrils. However, there are a lot of differences between cardiac muscle tissue and skeletal muscle tissue. For instance, cardiac muscle cells are uninucleated. They only have one nucleus. And they are much shorter than skeletal muscle fibers. So they don't run the full circumference of the heart. They are just in small chunks. And also, cardiac muscle cells can branch, so they don't necessarily have to be a column in shape. And another important thing is we can see structures called intercalated discs in cardiac muscle tissue. So intercalated discs are dense protein structures found between adjacent cardiac muscle cells. So these are the desmosomes, the gap junctions that are connecting the adjacent cardiac muscle cells to each other. That's what an intercalated disc is. And finally, the cardiac muscle tissue has autorhythmicity. It is involuntarily controlled. We do not have to think to make our heart beat. Matter of fact, the heartbeat can beat independent of the brain. Pretty crazy, that. All right, and finally, we have smooth muscle tissue. Smooth muscle tissue lacks striations, no myofibrils. Smooth muscle cells are uninucleated, one nucleus per cell. They are kind of spindly in shape, so they taper off at their ends. So thick in the middle with the nucleus, tapering toward the ends. Smooth muscle tissue is also involuntarily controlled. So we do not have to consciously think to get smooth muscle tissue to contract. It does need some sort of impulse to trigger it, but it does not need us to consciously think about it. And smooth muscle tissue is found in most of the hollow internal organs. So in the walls of hollow internal organs like the stomach, the intestine, small intestine, large intestine, the urinary bladder, the urethra, the esophagus. There's a lot of places where you find smooth muscle tissue. Blood vessels. So if there's no myofibrils, how is it that smooth muscle tissue can contract? Well, it turns out that it does still have thick and thin filaments, but instead of being in that dense column of protein, instead it is more disperse throughout the cytoplasm in the smooth muscle cell. And so the thin filaments end up being attached to structures called dense bodies that are scattered throughout the cytoplasm, and this attaches the thin filaments to intermediate filaments of the cytoskeleton. So the thin filaments are attached to structures, dense bodies that are attached to the cytoskeleton. And then, of course, the thick filaments can then connect nearby thin filaments together. And this means that the thin filaments are attached to the cytoskeleton, attached to the cell membrane or sarcolemma, and the thick filaments are connecting the thin filaments to each other. So we are getting a sort of mini sarcomere type structure. Then when contraction is signaled, the thick filaments will pull the thin filaments toward the center of the thick filaments. This will pull the cell membrane, pull the cytoskeleton, causing the smooth muscle cells to get shorter. Again, when they're relaxed, everything's as dispersed as it can get. When they contract, it causes the muscles to get pulled together, to get shorter. All because of the thick filaments moving the thin filaments closer to each other, with the thin filaments attached to the cell membrane and the cytoskeleton. And here is just a figure showing similarities and differences between the different muscle tissues. You might love it. All right, overuse. Overuse can cause tearing in muscles. Minor overuse can allow a person to recover relatively quickly, not a big deal, just little microscopic tears that can be fixed. Continuing to overuse a muscle after it's become very sore, though, could lead to more significant damage. Muscular dystrophy. Muscular dystrophy is a series of disorders, a collection of disorders, in which the muscle tissue is being destroyed and replaced with connective tissue. So progressive disorder, over time, the person loses muscle function. And then there's also myofascial pain syndrome. Myofascial pain syndrome is inflammation of the connective tissues in a muscle organ. It can be any muscle, doesn't have to be in the face. And what happens is when light pressure, someone gently touches that muscle, it triggers severe pain. So severe pain from a light touch because of inflammation of the epimysium or paramysium or endomysium. That is it for this part.